Uh, we're going to see about high throughput virtual screening. And in this, it's a extension. Think of it, this topic as an extension of molecular docking. And molecular docking is an integral part of virtual screening. Now, when we say virtual screening, is it exactly the same as molecular docking? Yes. Is it different from molecular docking? Yes, in the way the strategy of screening molecules is concerned. Okay. So how do we go about doing this, and how do we look at the limitations? What things has to be considered? What things you have to be like what kind of controls you can keep? It's like an experiment. This virtual screening, even though we do it online or we do it offline on the computer in a workstation or a cluster, think of it as an experiment. And just like an experiment in a wet lab where you will consider controls, you also need to have a plan. You need to have a hypothesis. You need to have a good setup, a strategy to think about how to go about doing it. And if you have you have been attending three previous lectures of mine, just like what I have been telling in the other three lectures, all these strategies are a basic guidelines, and they change from system to system that you are studying. And when it is a different system, a guideline given or a steps used may not be applicable to another system, and that will change. And that depends on what is your host target, what is the library that you're screening, and so on. Okay. So when you take the number of years of drug development, you see in the market, one drug has been released. At the level of the number of compounds that is taken to get to that one drug, you will have a huge funnel and a narrowed down funnel where there will be a lot of failures. So you in the discovery phase, and then the development phase, and finally in the market phase, anyone most of you might have seen this, where the number of years taken for a drug is about 15 to 20 years. And the amount of money that goes is about like crores and crores of money, like 500 to 100,000 million dollars. So, and the company, usually these are pharmaceutical companies that have invested this much amount of money in a drug. When they file for patents and then they are able to market it, they have only about 20 years to recover all of this money. So most of the time, philanthropic causes end up giving more money to compensate the industry. Industry also raises money in many other different ways. So you have to test many, many, many different compounds, many, many different classes of compounds to lead to this one particular drug. When in the case of virtual screening, we are somewhere here. We have like million, billion compounds which we need to shortlist to about 500 or 5,000 or maybe couple of hundreds that we can test it extensively in the lab. Now, before computational power or before 1980s or even 1970s, what happened was you would not take these many compounds to test. You will probably start the discovery process here. Like how I'm talking about, like, let's say a particular plant. Okay, if you look at Taxol, okay, Taxol is a known toxic compound from the plant, and whatever molecules they could extract, it was a couple of hundreds. It wouldn't go beyond to a million or a billion compounds. But as computational power increased and as databases came up, as the algorithms got developed, we were able to get more compounds that we can screen. That means all those compounds were being sold by the chemical companies, and the biggest supplier of chemicals is Russia. They have special chemical zone, like special economy zone in India, where you have hundreds and 200s companies where they are all synthesizing different chemical entities, and they have a library. Now, if you imagine a 96 well plate, a micro titer plate, you have 96 compounds, or at the maximum 100 compounds you can test in one plate. Imagine you are screening 100 plates like this. Now, in a day, you can screen probably thousands of compounds. If you have 1 million compounds to screen and you have to find which one among them is a best hit or a good hit, which has some slight indication of activity, you'll have to go through a whole amount of resources in purchasing the compounds, in setting up the assays, expressing the protein that is required for the assay. So there's a huge amount of experimental wastage that happens. So the usual way of screening is experimental screening. Now, since computation came up, you can test a lot of compounds in the dry lab or in the computer, and then you can start with a new testable set of compounds that you can test it in the experiment. So you save money, you save energy, you save time, and then manpower. So virtual screening is also called as hit identification. Now, these are the two terms that is shown in the previous slide here, hit and lead. Okay, what do we mean by that? I'll tell you. So all the compounds, 1 million compounds, let's say you are screening, or 1 crore compounds you're screening, each is a potential drug molecule. Okay. Now, anything that shows a promising uh, 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 assay or activity or some kind of binding, we call that as a hit. So many of them may not be hits, they are non-hits. Okay. So among those hits, there might be few that might have a good binding energy or a good interaction that we will see. And those molecules are called as leads. Now, this lead molecule is cannot be used directly as a drug, because that lead molecule has extra advantage over the other molecules that has been screened so far. You have to optimize the lead to find the best possibility in that particular molecule. And I'll in the subsequent slides I'll show you how one does that. And you have to screen again those possibilities, and then we call that as focused library, and then we optimize it and then see which one has a much better activity. So if your hit gave millimolar IC50 values, your leads might give micromolar IC50 values. But drug discovery process, any molecule that should be considered as a drug, you need nanomolar IC50 values. You need to have very low quantities of the drug to give a huge effect. That is the desired characteristics of a drug. So think about it. If you have a headache, you take a paracetamol, it is 500 milligrams. Now, 500 milligrams is the dose that is enough to overcome a pain by blocking the pain receptors for a period of 3 hours for a normal human being who weighs about 60 to 70 kilos, or let's say 75 kilos in weight. Okay. 500 milligrams for that. Now, the tablet that you take is not is doesn't weigh 500 milligrams. The tablet weighs about 1 gram or so, because there are carriers that are bound with the active component, and that's what is injected, or you can take it orally. So the nanomolar composition is the desired property for a drug to be effective, to give a huge effect. So 500 milligram of paracetamol is enough for me to overcome a pain for a period of 3 hours. So a normal dosage for a human recommended is you should not take more than 3 paracetamol tablets in a 24-hour period. More than that, it leads to hepatotoxicity. Your liver goes for a 6. So those things are not considered at the point where virtual screening happens. What is being considered is that whether you are finding a molecule that can lead to identification of a lead molecule that can further identify a drug molecule. That is what we are trying to see. So when, like is in these 3 bullet points, if there are 3 steps, that means screening of those potential molecules, you have to do it 3 times. And you cannot spend 3 experimental setups to find that, because that is expensive. Any company would like to reduce the cost and increase the profit. That's the business model. So if we can reduce the cost by doing it in a virtual way or in a computational way, that's called as virtual screening. And so virtual screening can also be applied even in these steps, but we don't call it with that name. We call virtual screening as the initial set of screening of potential molecules that can lead to a drug as virtual screening. There are a lot of advantages which I have right now I have listed. You reduce cost over time, you reduce the number of animals used, it improves the success rate. There is some kind of improved understanding. And this is what the other day someone asked me about, what is this rational drug design? You are going with a hypothesis, you are going, you know the structure of the ligand, you know the structure of the protein. And when we know both the structures, you can do a rational drug design. And that understanding is improved. You have a much better knowledge of the molecular mechanism that has. So you save a lot of time. So within nowadays, uh, whatever virtual screening is done for like maybe 1 month, in the traditional way, without computation, it would take about 2 to 5 years to complete the same task. So you're saving millions and millions of dollars. So in the computer-aided drug design, virtual screening is done for this structure-based drug design where we have protein ligand docking. And in the ligand-based drug design, you can do the virtual screening using a QSAR or a pharmacophore to identify ligands that look like an active component. And also you can do it in the ADMET screening. Okay, so virtual screening can be done in multiple ways, but we are mostly focusing on protein ligand docking. So in a structure-based drug design, as I said in the yesterday's lecture, the good analogy, but not a great example, is like an assassination attempt. So just like how assassination happens by looking at the uh, uh, the routine of a person, finding out which particular moment of time in that 24-hour cycle they are most vulnerable. The person who wants to assassinate, they assassinate that person. That's usually the time. You will thousands of examples, right? John F. Kennedy was assassinated when he went without a when he went in a car without a hood. So, so what are we trying to assassinate here? As I said, it's a bad example, but a good analogy. We are trying to prevent the protein's function from happening, and that is what most of the time drug discovery is. The lead or the molecule is supposed to inhibit the function of the protein. Why do we want to do an inhibition? Why can't we go the opposite way of enhancing the protein function? Is because you have in a pathogen or in a drug receptor, like let's say the pain receptors, the the molecules that signal the pain, they go and bind to those receptors. If those molecules don't bind to those receptors, or something is blocking those molecules to bind to those pain receptors, then the pain response is not given to your system and you don't feel pain. So you're preventing the protein or you're preventing the molecule from doing its function. Simple as that. So whatever structure you have, the more you know about the structure, for which you need to have the crystal structure or the experimentally derived structure. There have been attempts where you can do virtual screening with a structure that is computationally derived. Then in that case, You can give the excuse or you can give the reason that it's not crystallizable, that the structure is too big, it's like 3000 amino acids in length, we can't solve the structure. So you have to validate the computational structure. One underlying thread that you will see in any of the computational based setups or computational based experiments is that you will have a high false positive rate and you will have noise and error incorporated in your results. How do you cross verify whether that is truly a signal and not a noise? And that is the reason why even computationally determined structures like the ones that you get from AlphaFold, homology modeling, or Modeler, or Discovery Studio, which is shown here. How do you verify that this is the structure that you see in reality, or is it this structure closer to ground truth? So you have to think of just like how in a wet lab experiment, you would do multiple other orthogonal experiments, cross verification to find out the real activity. You have to do other verification of this structure to find out that this is the lowest energy structure. This is the native configuration of the protein to which you will want to find the binding pocket. So this is the first and most crucial step in virtual screening. If your input structure is not validated enough, if your input area where you want to screen is not validated enough, then you are adding noise to your results. You are giving a high false positive rate. So active site identification itself is a separate topic where after you have identified the target, which again is a separate topic, you want to make sure that the site at which you're going to do the virtual screening is in fact the functional part of the protein. Okay, and that is something very crucial. And these are all the steps that would have happened before you actually do virtual screening. So virtual screening, as I said, it is molecular docking. So you find the best matching between the target protein and the drug candidates, and you have many different tools. Yesterday we saw about Autodock, and the thing about molecular docking is that you're remember that you're not asking the question where, you're asking the question how. So you're not asking where does this molecule bind in the protein, how does this molecule bind to the protein? That is what is being answered. So if your question is where, then you probably have to answer the question of the active site identification and then come to the virtual screen. Again, this depends on the system. So if you have a crystal structure of a complex, then 50% of the problem is solved. Okay. So in the first type of virtual screening is where you have a structure-based virtual screening where you use a 3D structure of a protein. Either you take the protein structure from an X-ray crystallography experiment or a cryo-EM experiment or NMR or X-ray free energy laser or computational structure. AlphaFold, I'm pretty sure all of you have heard about it, that it got the Nobel Prize. Denis Hassibis and John Jumper got the Nobel Prize for AlphaFold, and many news agencies carried the news that Nobel Prize was given to the computational method that solved protein folding. No, it didn't solve protein folding. It absolutely didn't, but it sped up the process of finding a three-dimensional structure of a protein in the most reliable way. And the method was not something new, but implementation to a huge amount of data was something new, and that only DeepMind could do it, which was uh, a company of Google, because Google has huge data storage, huge infrastructure to do this kind of research. Microsoft, Facebook, they are also doing different types of research on all these protein structures, and they are also into drug discovery process. So once you have the 3D structure of an AlphaFold protein or by experimental methods, you have to like you do the docking and then you find out the interactions. You can automate the interactions using fingerprint. or coming up with a pattern. You can do pharmacophore mapping. You can do binding free energy estimation. All these are done post docking or during docking to find um, how it can affect or how it can possibly bind to the target. So you can do this type of a virtual screening only when you know the target structure and the model structure is reliable or not. If it is not reliable, like many of the AlphaFold proteins, the problem is people started trying it out for their protein of interest, like a cancer target or some protein that they have been trying to understand in different way. And it came up with a spaghetti. So basically, it was all loops. And that reason is that majority of the proteins are intrinsically disordered, and intrinsically disordered proteins do not have one native state structure. They have an ensemble of structures, meaning at the near native state in the energy landscape, you will have multiple structures that can have the same energy, but they will keep on fluctuating between two or three different conformations. And when they do that, they will be highly flexible. So those type of drug targets are very difficult to model. And the second reason is that even if you have a target structure known, like through X-ray or cryo-EM, because they have some intrinsic flexibility at certain parts of the protein or parts of the structure, they'll be missing. So the data will not be that. So you have to build those missing residues. This also defeats that the earlier hypothesis of if you have a gene that codes for one protein, the protein uh, is doing only one function. That is also disproved in recent years. You have proteins that do multiple functions depending on the context. So you have moonlighting proteins, you have proteins that are like Dr. Jekyll and Hyde. They do one function in a particular part of the cell, they do another function in another part of the cell. A protein can have multiple functions depending on how many domains it has. Each domain can have its own function. So complexity of the target structure, or which what type of structure you're going to use, again makes a different strategy. You need to come up with a different strategy to do virtual screening there. So in the ligand based drug design, you have molecular shape matching. So let's say one particular ligand has type of a property, so you don't know the receptor structure. Let's say arthritis. Arthritis is a systemic disorder. You have multiple targets that are affected, and you have symptomatic relief through a particular compound. So you want to find out a much better drug for arthritis, which can ease up the pain, or it cannot just be painkillers. So in that case, you want to find the receptor that it binds to, but then the receptor is not uh, easy to determine. It's most most often they are GPCRs, which are difficult to crystallize and difficult to solve. They are membrane-bound proteins, and so you have a library of molecules which are known to have similar effects of preventing pain in arthritis. Then can you come up with a model for another molecule that shares the same properties as the known molecule, and then you can design a new drug? That's also virtual screening, but without the structure of the receptor, and that's what most of the ligand-based pharmacophore designs undergo. So you use known active ligands to find similar molecules. You can use pharmacophore models or QSAR models, quantitative structure activity relationship. These are basically regression models. And in recent years, we use machine learning approaches to predict what type of molecules are closest or similar to active ligands that have the same function as the known structure. So when to use this, you have no structure available, but active compounds are known. You have a library of compounds. They are proven to work. but you don't know where they bind to. The molecular mechanism of it is not known. So then the third way is where you have the admit based. So you look at a complex set of data. You have animal data, you have structure activity data, you have clinical data, you have literature data, you have in vivo laboratory data. Combining all these heterogeneous sources of data, you want to do a data mining approach and then find out what type of ligands or what type of molecules can be identified from a library. So that is the data mining approach. So this is very frequently used, very useful to find out whether shared features of molecules can be tapped into to identify leads, and that can probably lead to a drug. Many different tools are there, many different websites are there. This is what we saw yesterday. The databases are a lot. There are a lot of tools, a lot of databases, a lot of approaches. What is one take home lesson at this point of time is there's no one glove that fits all. You have to tailor your approach to that particular system, to that particular target, to the particular outcome that you're desiring in a virtual screening approach. Okay. So, and of course, you can have an hybrid approach where you have a ligand based virtual screening. It's you identify first and then you go to a structure based refinement. You can use AI based scoring, RF diffusion, evo bind approaches, which is for peptide design. And then when you try different hybrid approaches, you come up with a consensus, meaning two different methods, two different algorithms give the same result. That means your confidence in that result might be better. So your prediction is higher, and you would want to reduce the false positive. So consensus based approach is one way I also used in my study for identifying a lead in lead against dengue viral envelope proteins. So we used three different methods and then found out which molecules are predicted better by all three different methods. So that is a consensus-based prediction. Now, when we do when we do virtual screening, the input of the target and the input of the molecules that you're going to use, these two are pretty much uh, where you have to spend some thought and spend some time in identifying which one you have to go for. Usually, you take one target and you have a library of compounds to do molecular docking with. That means you're in a pharma company or in a drug target identification pipeline, your target identification team will come to you and say that, okay, this is the target we have identified. Now do your molecular docking and give us, and then send the results to the next team. So you have very less idea about how they ended up identifying the target. Of course, there will be some information given about the target identification, but then there is not much control over going back and redoing the target identification. So in that case, your task is probably just to screen the compounds. And when you are screening compound, high throughput, the word high throughput means that you are selecting a huge library of compounds. So you want to start with very high diversity in terms of chemical compounds. When we talk about diversity, what do we mean here? We mean different structures, different entities, uh, different properties of the molecules, different variations. If the molecules have chirality, then you can have isomers, you have tautomers. If the molecules are highly flexible, then you want to have a library made of different rotamers. So all this information is required when you are starting with a library selection. You can go for many different types of libraries. Commercially available libraries from Zinc or ChemDev. ChemDev is a company that sells a lot of chemicals. It's still in business. You can even get natural product libraries, which are specific compounds synthesized or extracted from marine organisms or from plants that uh, are not easily available, cannot be synthesized by a medicinal chemist. They usually are highly complex, that it's a multi-step process and it's more than 20 or 30 steps to get the final product. So then also you can do fragment library. You basically want to identify um, functional groups, or not the entire compound, but parts of the compound that show a good activity. And then maybe from that you build much more focused library from that. You can also go for custom build libraries and so on. So what usually companies do in commercial libraries is that uh, you don't know which molecule you want to buy from them, and they will sell it as 1 milligram, 5 milligram, 10 milligram amounts, and they will be in powder form, and they will sell it at 99.9 or 95% purity. Each of those compounds costs 100 $200. Okay, they are expensive because they are chemically synthesized. So before you order from them, you can download or you can get the library data in a virtual format like the SDF format, multi SDF or multi smile format, and then you do your virtual screening, and then you order to the company, okay, these are the compounds that I want from you. Okay. Or even if you're not going to buy it, you can create your own in silico mega libraries using multiple tools are there. You can create your library. So basically, the first step in virtual screening is library selection, and you want to create a huge set of molecules that you want to test against one target. Now, just like an experiment, you want to have proper controls. So using decoy molecules is an essential step in virtual screening. Now, what are decoys? Decoys look like an active component or look like a drug molecule, but actually uh, they are not, and they are duds basically. Okay, so decoys are computed based on similar physical properties but different chemical structures. So this text I took it from a website. I'll just show you. So none of these decoy molecules will be active in experimental setup. Basically, these are negative controls. Okay, so they are non-binders, and you can incorporate that in your virtual screening. The reason is, if you are finding a molecule that is a good binder, it has let's say minus 10 kilocalorie per mole. What was the binding efficiency of a decoy molecule to the same target? If it was minus 4 kilocalorie per mole, then okay, you have got probably a good binder there. But if your decoy is minus 20 and the one that you found was minus 10, then probably that's not a good binder. It may have good interaction, it may have other properties, it may have a perfect Lipinski's rule of 5 and everything, but it did not. It's a completely false positive, which means that they don't perform as well as the decoy molecules. So dud, or it is pronounced dud e, is derived from zinc, and you can even purchase these decoys, and you can test it out and get a negative control in your experimental setup. Then what is a positive control? Positive control is a molecule that shows the effect, or it can give rise to the correct signal. So, for example, let us say you are designing antifungal molecules and you have a library of antifungal molecules, and then you are having a decoy molecules which look like antifungal molecules, then that's the negative control. So, in that case, the positive control will be known antifungal molecules like amphotericin B or some other antifungal drugs. Okay. So this is the website, a database of useful decoys enhanced. So DUD-E is a revised version of the previous DUD database. So you can, this is the paper in 2012. It was, again, Irwin, this is the people behind this is those who were involved with the doc algorithm. So this is how you can generate your own duds. So you can give your input smiles, and then you can generate how many ever molecules you want to uh, try that looks like it, but basically they are mostly inactive molecules. Okay. So at the core of this high throughput virtual screening, you have different approaches to molecular docking. And the approaches to molecular docking depends again on your system, depends again on what the question is and what is the aim of this entire drug discovery process. So you have either you want to do rigid docking or you want to do flexible docking. Again, it's a question that you have to ask, and that depends on your computational resource, your host target size, the library size, and what is the time that's available, and so on. Whether you want to do a blind docking or you want to do a site-specific docking at one particular step of the virtual screening, that again depends on your computational cost, your time, and how much and what is the outcome that you're expecting out of a blind docking, and what type of scoring functions you want to use, what type of tool you want to use. So do you want to go for empirical scoring functions or force field based scoring functions or some other type of scoring function? It depends on whether you have the resources to buy those scoring functions, or you are going to use a consensus based approach. So you try different scoring functions and see which one is giving, or if all of them are agreeing to the same outcome. So whether you're doing one step process of screening the molecules in molecular docking, or you're doing a multi-step process. Okay. Usually they do multi-step process in a virtual screening. So if it is a multi-step, then how many steps you're going to incorporate? What is the strategy behind it? So all these questions you have to ask in a virtual screening before you start. And you need to have some kind of a estimate about how much time it will take at each step. And in Autodock yesterday, we showed 10 dockings per molecule. And the setup was in the Lamarckian genetic algorithm was medium. If you switch it to short, it will not probably take 51 minutes for each molecule. It will probably take 2 minutes. If you switch it to long, which means the number of populations or the conformational search is more exhaustive, then it will probably take 9 hours for each molecule. So which one will you go for? It's obvious that when you have 1 billion molecules, you're not going to spend 9 hours for each molecule. You want to, as I said in one of the earlier lectures, you want to fail fast, which is the motto of industry, that you want to identify or you want to eliminate bad binders as quickly as possible. So you have different setups, you have different tools to do that. Even within Autodoc or even Doc, you can change the algorithm, you can change the parameters to do different types and then see whether you are filtering it out. So Glide, Gold, they are commercially available. Doc, Autodoc, Vina, Rosetta, Ligand are not commercially available. So again, but if it is an industry, then there is an industry license that you have to buy. If it's academic, you don't have to buy the license. You have to apply for it and they give it for free. So you can do rough docking or detailed docking. Again, it depends on what time and how many compounds you want to screen. So you just want to see eliminate bad binders, you do a rough docking. You want to see among the set of 5,000 compounds, if I'm able to find any that is pretty interesting or not, and I don't have a time limitation, I don't have a computational limitation, I can keep on doing detailed docking for all 5,000. But usually we don't do that. Because in detailed docking, you just don't do docking for multiple conformational search. but you do more other detailed analysis, like for example, thermodynamic properties. You calculate the entropy, enthalpy properties. You calculate the um, uh, KD value or KI values through other tools. And so the time taken for one molecule might be more compared to the rough docking. So there are different levels at which you can do. Now, one of the things that you'll come across in any virtual screening paper is that how much of the true positives were recovered at the end. So the rate of identifying the true positives. So that is the positive control. So was it improved after multiple steps of the virtual screening? And the enrichment factor. So here it is A by N, where A is the number of active compounds in the top N compounds. So let us say you have 1 billion compounds and you have screened, you're looking at the top 1,000 compounds. How many of them are actually the true active in nature, true positive? Okay. And that number is divided by an uppercase A by N, where uppercase A is the number of active compounds in the entire library of n compounds. Okay, so that is 1 billion, right? So the sample that you are taken to screen further, if let us say it was only 10% were active compounds, to the next step, do you expect those 10 compounds or 10% of those active compounds to be enriched further and remove more bad binders? Thereby, it becomes like 80% of the entire final set of molecules that you're going to screen. That is the enrichment factor. So, you the trade-off in virtual screening, it just so happens that when you add more filters, when you add more layers of constraints, like let us say, for example, first you start with Lipinski's rule of five, then you say log p value, then you say high C50 value if it is known. The more you add, what happens is that there is a trade-off of removing a good binder in one of those filters. And that trade-off is okay. So I hope you understand what trade-off means. So trade-off is nothing but what are you sacrificing to gain something? Okay. So how many of the good binders are you losing at the initial filter to have a good enrichment of true positives at the end? Okay, that is the trade-off. So if you're losing like maybe 10% of good binders at every step, that may be a pretty high loss, high trade-off. If you're probably losing only 1 or 5% of the true positives at every filtering step, or different steps of the virtual screening, then that's probably a better trade-off. Okay, so again, it depends on the system. Again, it depends on how many molecules you're going to screen, how many molecules you start with, and so on. Okay, so but these things are not there to make your life difficult when you do virtual screening. These things are there to ensure that the molecules that end up being developed as drug at the end and being that for which a patent might be filed and you might end up getting um, a license, they have to have a good validation. You need to get a good confidence that it's a good drug, good binder. That's the reason why all these things are there. Okay. Success and failure, again, based on the positive score, positive control and the negative control. Most of the time, if you have a crystal structure that you have a known drug molecule and you're finding an alternate drug molecule that binds to the same target, how is it compared to that existing crystal structure? So the best scoring mode and the closest configuration. So the quantitative approach is to calculate the root mean square deviation, and you want to find out a root mean square deviation between two molecules uh, with the known and the new one as close as possible. So RMSD of less than 1 or zero point 0.5 angstrom is desirable. If your RMSD is 3 angstroms, 4 angstroms between the known molecule and the predicted molecule, then that's a very bad result. because it's completely different mode of binding, which is not probably what is happening in reality, because your crystal structure is more closer to reality. And again, understand the PDB structure or the crystal structure is an average model among all the different conformations that is present. So it is it is a model. Again, it's a model. Okay, so how closest is your prediction to the model? Then assigning lowest energy score to the correct binding mode is more difficult. This I think I covered in the first lecture. So the energy score, it depends on the scoring function. Every scoring function will give a different value for the same molecule. Then what is the correct energy score? You don't look at the absolute value, you look at a trend. You look at in relative terms. So that is why the positive and the negative control is required in a virtual screening. And then ranking order. So you have two compounds. The difference between them is just the hydroxyl group. Nothing else is different. Everything else is the same. How do you rank them? The one without a hydroxyl group is ranked higher, or one with a hydroxyl group is ranked higher. So what is the criteria to rank them? Binding energy is fine, but if they show completely different binding energies, then does is the hydroxyl group that is driving the interaction and making this difference? How true is that? So those type of questions you have to ask if you are ending up with molecules that are highly similar and having very close binding energy values. And that is the whole reason why you want to start with diverse set of molecules, and you get different chemical entities at the end to test. And as I said, having a control setup is crucial. You need to know what you're looking at, because in computer science, and because this is done through a computer or through computational way, in computer science, the common fallacy is GIGO, GIGO, garbage in, garbage out. So if you don't know what you're looking at, then most of the time you're just looking at noise. So you need to know what you are trying to find. And unlike biology, computational science output is determined. In biology, we are trying something and then we see if it works. If it works, then that's what the outcome is. But in computational setup, the input is defined, output is defined. So you need to look out what type of molecule you are looking at, what type of molecule you are expecting. So the variables, the more number of variables are being factored in, the better will be the success. And most of the time, virtual screening is an automated process. You run the simulation, you run the docking. You have 1 billion compounds. Each compound takes 2 minutes, let us say, to run. You create a shell script, and then you take one molecule, do the docking, and give the output, take another molecule, do the docking, give the output, like automation. Okay. What happens after that? That is equally crucial compared to the setup and the strategy that you think before you do the molecular docking. So you have to remove the highly uh, uh, binding poses that are energetically higher. You have to find out if you can do some type of clustering in terms of RMSD, or in terms of the orientation, or in terms of binding energy. You have to do visual inspection. Most of the time people forget to do visual inspection. This is structure based drug design, so you have to look at the outcome. You have to look at the molecular viewer, and find out whether it is binding in the proper way. Molecular docking tools sometimes take 2D molecules and then do the docking also, but then 2D is not what we see in reality. There are 3D molecules. So uh, did by mistake did you input, or did you do a docking of 2D molecules? It's very easy to find out, because when you visualize it, they'll all will be planar. Everything will be like a plane. And so that means it was a 2D structure that was given as input, which is not at all good. So then gives free energy rescoring. That is very easy to do these days. So earlier it used to take a lot of time, computation time. Now MMGBSA tools, you can find out the delta delta G values very easily. Then you can do the interaction fingerprints. You can find out what type of interactions are made. You can automate that fingerprints as well. And then you can do, uh, you have to overcome some type of a bias. The bias that is introduced here is, let us say in the control setup, your positive control, you didn't remove the origin. Okay, so this is a crystallographic term. So origin is nothing but, let us say, this particular, I have an example is this VIX. Okay, so this object, okay, I can keep the origin at different points, because because the coordinate system that we use with molecules when we do virtual screening is an external coordinate system. It's not internal. In an external coordinate system, when I say 3.9.6 in the y-axis, 2.1 in the x-axis, y-axis, and then 5.9 in the z-axis, that means it is present from an origin which is 0, 0, 0 to 3, 2, 1, 5, and somewhere here. So this is my origin, and if you look at the video, this is my origin, and this is where it is. So in the positive control, the crystal structure that is bound with the protein, if you have not changed the origin and you are just removing the molecule from the crystal structure and then putting it back, the coordinate space, the external coordinate space, preserves this origin. So if to remove the origin, you have to scramble this molecule. You have to uh, take it in a different conformational space or change the origin. And then when you do the docking, are you able to recapture the crystal structure? And that will tell you whether your molecular docking protocol, your settings was good enough or not. So you can try different methods, you can try different approaches, and then see whether all of them give the same results, the convergence. And that is what we call as consensus scoring. So consensus scoring is because even for a simplistic system as like a hydrogen bond, okay, every scoring function has only so much empirical data to calculate with, and there are errors in that, and the error propagate. So when you do a consensus scoring, you are trying to remove those kind of biases, so that will make it your result more closer to reality. Then ADMET prediction, drug likeness prediction, which I'll not go into detail, but this is also a type of filter that you can do either at the post-docking analysis or before you do the virtual screening. And there are many tools to do that. And then one of the often validation that is done after the virtual screening is molecular dynamics simulation, where you try to see the stability of the binding. So technically, I say this statement, and this statement is true in a computational sense, that you can dock a cow with a buffalo. That doesn't mean that's what you see in reality. So a cow and a buffalo may stand nearby to each other, but they not necessarily will be like, hugging each other and having an interaction. Okay. So when you do molecular docking, you can predict, okay, this particular molecule binds to this particular target, makes an interaction, has a binding energy, fine. Does it like binding to each other? Okay. How do you find that out? And the way you can do it is by molecular dynamic simulations, where you find whether it's a stable interaction or not, whether they like to be with each other, or whether they drift apart within a very few nanoseconds. So usually the practice these days is to simulate the complexes that you get for at least 100 nanoseconds and in triplicates with different starting conformations, so that you have convergence and replicability. And then you look at the different quantifiable factors like root mean square deviation, hydrogen bonds, root mean square fluctuation, and then look at the Poisson-Boltzmann solvation uh, parameters and all that. All these things are done to verify whether the true positive that you identified is indeed a true positive and whether that drug is indeed binding to that place that you are trying to. What you have to understand in reality, what happens in a cell or in a real environment is that protein and the ligand, they will come closer in a space and they will make an interaction, and this ligand will not stay in that particular place for a long period of time. It will dissociate, it will go and interact with another molecule and come back, or probably, or within this molecule it will try to roam around its surface. All these things happen very, very common. And because of this, the protein will undergo a conformational change. The protein will try to recapture the ligand and try to bind to it tightly if it is exactly looking like a substrate. Or if it doesn't like it, then the protein will probably throw it out, and it will um, not like binding to it. So when we do MD simulations, we try to find out whether we are looking at the ground truth. So there's a question in the chat box that says, what are the limitation of post-docking analysis? The limitation is that it takes time. You are trying to see with multiple analysis if the prediction is true or not. So time taken is the limiting factor there. Otherwise, you don't have any other limiting factor. You're just validating whatever you got from docking as true or not. So there are a lot of tools: GROMACS, Amber, NAMD, OpenMM, and there are even online servers to do molecular dynamic simulation. So most of the time, people will feel limited that you need to have workstation or a cluster to run it. But you can run short simulations in online servers as well. So in the high throughput virtual screening, what are the pitfalls and challenges? Scoring functions are not accurate all the time. There's a lot of research that's happening where people are coming up with better scoring functions. Even the commercial softwares, they will update their scoring function if they find that there is some empirical data that improves the prediction. So many of the time, the scoring will say, "Oh, this is a great binder," but when you do the experiment, you will see that it's not binding at all. Protein flexibility: when you do rigid body docking, protein is rigid, ligand is flexible, so you're not taking into consideration protein flexibility. Excuse me. So as I told you, it's computationally expensive. The limitation is you can't do flexible docking for 1 billion compounds. You can do it for maybe 10 compounds or 100 compounds, not more than that. You will lose a lot of time. False positives dominate high throughput virtual screening in any drug discovery process, and that is where a lot of time gets spent to remove the false positives. So whatever you think is a good binder may not be a good binder at all. And of course, commercial libraries, they will oversell certain type of molecules because they are easy to synthesize. They are produced in kilogram in their factory. They will not be able to produce many macrocycles, many peptides, many natural products or isomers. And so the library that you have picked up or that library that you're studying has some limitations also. Sometimes the molecules present in the library may have poor admit, or they may not be able to synthesize at all. And this is very common with natural products. Natural products cannot be synthesized in the lab. You have to get it in from the original source, and those are not cheap, they're expensive. Future trends, you have deep learning based docking using AI, machine learning, where diffdoc, equibind, or these tools that they do use deep learning methods. There are different type of generative models for de novo design, like diffusion models that I talked about. So recurrent neural network models. So they come up with new chemical entities, and it's up to the medicinal chemist to see whether they can be synthesized or not. And there is reinforcement learning or multi-property optimization. All these are new trends that are coming up. There are many new tools that incorporate AlphaFold type of prediction along with the protein. You can incorporate nucleic acids or metals along with the protein and then do the docking. Okay, so there are many, many new things coming up. Then integrating with multiomics, that's again happening. So we know the protein interacts with some other protein or interacts with another metabolite. The amount of protein that's present in the cell, the dosage required for the molecule to bind to the protein, all these type of data will enhance the virtual screening approach. And precision medicine is where we are heading towards. Very by the time we are getting old, we will see a lot of precision medicine happening in India as well. So there are a set of populations which harbor a specific SNP, and the drug will be specific only for those set of population. And in the entire Indian population, there may be hardly 1,000 people who have that SNP and who are suffering from that disease. So in that case, you are designing a drug only for those 1,000 people, or you're using. So because it takes about 15 years to develop a drug, for a new drug to bring it to the market with all clinical trials and all. You might have heard about drug repurposing, where existing FDA approved drugs, which were approved for a particular condition or a particular disease, you see whether they can be repurposed for something else, or where you can um, treat for another disease. Then high throughput, these days, as I said, it's computationally very cheap. You can get a cloud-based computation. You can do a GPU-based molecular dynamics simulation. Um, so you can do a lot more sampling. You can do a lot more ensemble docking, and then you can automate a lot of these pipelines in a high throughput manner. There's another question. When docking gives contradictory results across different sorter, how to resolve them? Again, consensus scoring is one way to go about. That's a trade-off. You see one tool is giving a good binder, that molecule A. Molecule A is not a good binder in two other tools. Then you don't consider molecule A at all in the next step. So you have to come up with a strategy. Again, this is something that is very system specific, meaning that it applies to the protein or the drug target that you're trying with. So, how many molecules can be purchased? How many molecules can be synthesized? What is the amount of um, manpower you have to test all that? And the second question is, can high throughput virtual screening be trusted for novel targets with no known ligands? That's entire entire out, aim of a high throughput virtual screening. So a particular protein, as I said yesterday, it's not an enzyme, it's a structural protein. So then in that case, you want to find out what type of molecules can bind to it and prevent that protein from doing its function as a structural protein. So an example is your common cold, your rhinovirus. They will be they have the capsid protein. So if you make a molecule that can prevent the rhinovirus capsid protein from opening the viral capsid and entering the host cell, then that's a great inhibitor. So how do you go about designing it? So the whole idea is that in a virtual screening is that you have a target, a novel target for which no ligand exists, right? And so you have to come up with a baseline. You have to come up with idea that okay, these can be potential targets. Then once you set up the baseline, that is when that becomes a positive control, and then you do more development with that. So how can you start? Like all of you are taking this course because you want to do some kind of drug designing approach, you want to start somewhere. So for student or classroom projects, go to the database, the structure database PDB or AlphaFold, select a structure, select a library, do the docking using Autodoc Vina, you do a admit prediction using Swiss ADME, visualize the docking using a visualization tool like PyMol, and then do a small simulation for 10 to 20 nanoseconds in GrowMax or Amber. Both are free tools, and then see whether you are able to understand the process. Your goal should not be to identify a drug. Your goal should be to identify how the system works, how the strategy works, what can be done extra apart from this basic setup that I just described to you for your protein or for your target. Zinc, this is the Zinc database. Zinc 20 is the latest version. It has about 230 million purchasable compounds in ready to dock 3D formats. Um, and For a new database that you want to develop, this is the paper that's there in the PMC library, PubMed Medical Center. So you can take from different companies like Cambridge, ChemDev, Maybridge, and then they will be in 2D SDF formats. Convert it into a particular format that you want to use it for your docking purpose. Autodoc needs PDBQT. That means you have to use either RTKit or OpenBabel to convert the format. Then missing hydrogens have to be added. If your molecule has chirality, then tautomers and isomers have to be generated. Then you have to optimize the 3D compounds. So in some cases, the 3D structures need to be chemically accurate. In that case, you have to use some kind of a force field based optimization for some of the compounds that you're starting from scratch. When you take it from zinc, you don't have to worry about it much. But then for some of the compounds, you probably need to use force fields for organic molecules. Once you have the database, then you can use a tool like Doc, Autodoc, Glide, Gold, and then you want to you want to have some kind of data management software there. Now, data management software will ask for metadata. Metadata is name of the compound, molecular weight, pH, PI, purchasable, the chemical ID in the company, the um, number of isomers that it has, the smiles information of the same, um, the chiral molecules part. And so there are a lot of data that you can add for one compound. And so that database management is required when you prepare a database. A focus library. This is a tool called iLibDivers, sold by Intelligent company. So let us say you have a molecule like this. So it will fragment this, like for example, into 6 different flasks. So this particular flask, you can replace the methyl group with something else. You can replace this 5-membered ring with something else, and similarly. So you can think of all the possibilities that can be generated for each of these flasks. So it will create you a library of compounds for a starting structure like this. So that is a focus library. So you have, like, you can even start a library where the substructure match is uh, present. Like for example, these particular part of this structure is common, but the intermediate regions are changed between them. So that gives you a library of compounds from one structure. You can create your own focus library. Again, this is all in silico. How easy it is to synthesize, that depends on the medicinal chemist. You need to have a medicinal chemist in your team. So let's look at some successful examples where the structure-based drug design and virtual screening have been used. So for the non-small cell lung cancer, sirsotinib, the commercial name is Salcori, was approved in 2011. Then axinitab in 2012 for renal cell carcinoma, Zelboraf for the BRAF mutation. So for it was identified for the selective inhibition. The most popular one everyone will talk about is Gleevec or imatinib for chronic myeloid leukemia. But this was one of the very successful case study. But the problem right now is we have Gleevec resistant leukemia, and there's a lot of research happening in pharma companies like Eli Lilly, Pfizer, where they're trying to come up with the next generation imatinibs. And there is Ibrance or palbociclib, CDK4/6 inhibitor. CDK is the cell division kinase. So it for breast cancer. Then there is this paper where I picked up this table from, where you have different drugs that have been successful and the year that have been released. Some of them have been in clinical trials. Some of them are still under clinical trials. Then there is a whole bunch of antivirals for HIV, for influenza, for COVID, where computer-aided drug design, especially virtual screening, was used to find out which molecule is effective in binding to, like these two, the last two for COVID virus, SARS-CoV-2. And there are other examples where FDA approved inhibitors were designed over a period of like 30, 40 years. And lot more is happening now because we have much better computational resources, and the virtual screening part is getting more automated, more streamlined. There are a lot of companies that use AI based methods, and they frequently do a much more enhanced, very fast computer aided drug design at the virtual screening part to identify leads. The sooner you identify a lead, the faster it is. With that, I'll show you a demo of, like, let us say I took this protein. Are there any questions? Okay, so let's say I'm interested in hepatitis C virus, and I have this UniProt entry for this. Excuse me. This is a polyprotein. The entire genome is available in this particular entry. It is about 3,000 amino acids long, but I am more interested in one particular part of the protein. And so I go to the structure tab. I'm pretty sure all of you know about this. And you will see for that one particular structure, like this region from 1027 to 1207, you have this 200 amino acid length, which is present in X-ray crystallography data, present as a dimer. This a resolution of 2.9, and there are many structures for the same protein from hepatitis C. Now, if I want to create an antiviral drug for hepatitis C, then which particular structure should I select? That depends on which particular target um, that I'm focusing on and in the particular viral life cycle. Do I want to inhibit the viral entry? Do I want to inhibit the viral replication? Do I want to inhibit the viral maturity? Do I want to inhibit the viral spread? So again, the target identification is just like an assassination attempt. So which particular part of the life cycle in the pathogen or the virus is most vulnerable, or which is something that you can identify very easily. So you'll have different types of structures for different regions of this viral protein, because not all are crystallizable. Some are very hard to crystallize, some are intermediate structures. And then you can see as you scroll that there are a lot of modeled proteins. So you can, like, how well do you trust it? That depends on this particular model. And when you once you, let us say, through a particular strategy, again it's a system dependent strategy, where it's completely up to you what questions you are asking to identify which particular protein you want to select or which particular target structure you want to select. And then you can go to zinc and you can select a particular tranche. Let us say I want to download a particular set of molecules with a particular criteria. So it will load a matrix here in the tranche where you'll have a log p value along with a particular other factor, like let us say molecular weight. Okay. So you have log p here from -1 to 5, molecular weight from 200 to 500. So let us say I want to select this particular tranche where log p value is 3.5 and I want molecules with 425 daltons. Then I just select this many number of molecules from zinc. So you can have a different types of selection. You can even go to biological entries. You can look at minor targets or major target classes. You can search through them and then you can get the molecules that are known to bind to that. You can even go through catalogs and then search for a particular type of entry. Like for example, this is analytic on discovery. This is sold by that company. This is all these are different companies that have shared their data with Zinc. Zinc is the biggest compilation of, and then similarly, PubChem also has a lot more data in it. So you can depend. You can it depends on you what you want to search and what you want to go for. Again, decoys is a very good approach. You can generate your own decoys here. You can have decoys generated like through targets. Like for example, they have decoys generated for acetylcholinesterase, um, or there is a adenosine receptor where the decoys have been generated by hand. So there's a human behind this. So you can select what decoys you want to take for, or you want to generate your own decoys, or you want to go for a particular subset of decoys, like for GPCRs, okay, kinases, or generate your own, like the one I showed in screenshot. You put your smiles and then you upload the smiles. It will generate a decoy. It will take about a particular amount of time. So they give you up to 50 smiles, which is pretty good enough starting decoys to do a virtual screening with. Okay. Um, I'll stop here and I'll take questions.